One of the ways that we use these cages a lot is to do experiments looking at virus infection in honeybees. Um, and we do lots of different iterations of these where we might look at how co-exposure with virus and a pesticide affects survival, or how different diets affect bees' ability to resist or tolerate infections, or how behavioral changes might occur in infected bees to cause transmission to increase. And so, like I said, this is done quite a lot. And so here we're going to have a short video of Ed Shea, another graduate student who works in my lab, talking about an experiment he set up and how we put the virus inoculation into these cages and use them for lots of different experiments.
All
To study
right, so here
honeybees
you can see
is a system we
our
call in
cage
vitro
setup
rearing
down
or in
in
vitro
the
larval rearing.
incubator room, which
And
for
this is a
reference
protocol
is set
where
to
we
about
graft young
50% of
first
the
instar
humidity
larvae out of a
and between
colony,
33
just
and
like you would do in
34
normal
degrees
queen
Celsius.
rearing. But instead of grafting them into
The
queen cups and putting them
cages
into a colony, we
are
rear them
all
in
going
the
to
lab
look somewhat like
in an
this with
artificially prepared diet,
two feeder holes. You have
in
the
sterile
top
or
one
controlled
and
conditions,
the
where we
side,
can then
which can be
manipulate
supplied
different
with
things
whatever sugar
to
solution, honey,
change
or
the
pollen
environment that they're
the
exposed
experiment
to.
desires.
So we can see here petri dishes
The infection
set up with
process
just this
is
diet.
done
And I should
using
say it's
these little
artificial in the sense that we mix
weight
it up,
loops that you can see
but
in
it's still
some
based
of the cages
primarily
where
on
it can be
royal
removed,
jelly. So we
but
purchase royal
they can be
jelly.
left in just as
There's
well
nothing you
during
can
the course
rear
of the
bee
experiment.
larvae on that's not royal
For
jelly based
purposes
that I
of
know of. But we then
delivery,
can
it's
add
often
sugar
easier to
solution and some
straight
yeast
up
protein
label
and things like that to make it into more of the
the
worker
weight loads
diet, which is
and
much harder to
then just
collect than
insert
pure
them
royal
into the
jelly.
inoculum
And so we can
as the
take petri dishes
magnetic
like
induced
those
vehicle
here,
weight
make a bunch of little
load.
blobs of food, and then we can graft those really small first instar larvae out of the
After
colony
sliding in the
into these little
inoculum using
blobs of
these
food. We can just like very carefully
these
set them
easily
in so they
openable
float on there,
doors here,
and we can rear them to adulthood
the
totally in the
cages
lab.
are left
And so
for
we can
approximately
use
12
this tool to
to
manipulate
16 or
the
so
diet we
hours
feed
to
them, to
allow
introduce
complete
different
consumption
treatments
for the
during larval
inoculum.
development,
Currently, you can see
um,
the cages already have
and
feeders
um, and
inserted
see how they
along
respond.
the top hole.
And so, like I
But
said, we start out with this little blob of
during the
food,
inoculation stage,
and
all
we can
these
then
feeders have not
rear
yet
the
would not have
larvae,
been
and they look
inserted
like
yet,
this
and would instead include
after about 1 or 2
just
days in the
rather
food. You can see
centrifuge
that
tubes
food now
to
has
block
larvae in it.
this current
There's
iteration
about
is
5 per
no longer ready
blob, as we
fit
think,
today.
at this time.
Um,
And every day, we either go in and we move the bees from here to a new dish with new blobs of food, or for some experiments, we can refill the blobs by just squirting a little bit more of the food in there with them. As long as they remain kind of floating on it so that their side isn't covered, they can still breathe and they can still develop normally. If they get totally covered in the food, the spiracles on the sides of their body, the holes that they breathe out of, can become blocked, and they'll die. Here we can see another close-up of some slightly younger larvae that are still floating there on top of the diet. Here we see some where there's actually some variation in the size of the larvae. This is from an experiment where we weren't as tightly controlling the age from which we grafted the larvae. And so you can see some are clearly larger than the others. They're probably about a day older. Most of them are around the same size, but in the middle there you see a much larger one. So if we're doing an experiment, we would probably remove those ones at this stage because we don't want that age variation in there. Again, another example showing what they look like. They'll consume that entire blob of food in about a day, and we've calibrated how much food we put in to kind of match that. This is what it looks like if you wait until they consume all the food. They just are kind of laying there on the clean dish. You can see some residual food where some of them have not eaten it all. Another example where this is an experiment where we're rearing them in a really huge blob of food, basically one giant blob instead of many small blobs. But you can see how well they seem to do. You can have problems with rampant infection in there. But we've done a pretty good job of keeping these clean. One of the things I always kind of joke about is that we can do a pretty good job rearing bees in the larvae, but we're still the worst nurse bees in the world. We're worse than the worst nurse because we just can't do it as good as a real bee can do it. And one of those things that we can't do is do the level of grooming and cleaning that the bees do. You know, we can move the larvae from a sterile container to a sterile container, but if pathogens get introduced, it's very hard for us to clean them. While a worker bee would be inspecting these, grooming them. If a larvae was sick or dying, they would be removed by hygienic bees. It's much more difficult for us to tell if a bee larva is sick or dead until it becomes kind of too late and fungus starts to grow out of them. So we have to be really careful. Again, we can see another example of what that looks like. And so these bees are ones that are right about to pupate. They're really big. They're about to go into that pre-pupal stage where they'll start to look less and less like a grub and more like a bee. And here, after we can see what those will look like, these are bees who have reached what we call the pink eye stage, unsurprisingly, noted by their pink eyes. Before this, their eyes are white. You can see it looks like a bee. The shape of a bee is there, but they're totally white. These bees are slightly more mature. The eyes have started to darken. You can see a close-up again. And we can use these pupae for lots of different types of experiments. In this one, we were just seeing if we could get them to emerge. And then here's what they look like as they're getting much closer to being an adult bee. Here we can see they're darkening. The coloration pattern is there, but they still don't quite look like they're ready to emerge. And here's a figure from a paper that I published some years ago where we were rearing larvae in the lab, not on worker diet, but on a queen diet. And so we see on the right hand a control. This is a queen we reared completely in the lab. This bee never, after she was grafted out in the first day after she hatched, was never touched by another bee again. She was reared completely by me in a petri dish. And so she emerged looking obviously like a queen. The other three, the IRS Tor and IRS Tor, those are bees who are fed different treatments that blocked parts of their metabolism that prevented them from becoming a queen. And we found that not only were they smaller like a worker, but they also lacked a lot of the other queen characters. They didn't have large ovaries. Their mandibles were worker-like instead of queen-like. They had a barbed stinger like a worker, not a smooth stinger like a queen. And so this just shows the kind of output we can get from these in vitro larvae, that we can get them to become normal-ish walking, talking bees. And so we can compare how different factors during development affect what happens to these bees. Related to this larval rearing protocol, we use another method that we developed that also involves rearing brood totally in the laboratory. But this one is done a little differently and with some other purpose. A lot of those experiments I talked about earlier, where we use virus infections, um, for those we have to produce the virus particles, the viruses themselves that we use to infect the bees. Um, in most systems, in most animals, this would be done in a cell culture where cultured cells in petri dishes like human cancer cells or mouse cells, uh, in our case, we would use insect cells, um, can be used to produce pure live viruses. Unfortunately, there's not a viable virus-free honeybee cell line right now that's widely available. And so instead, we do it a little bit differently by harvesting a large number of well developed larvae and pupae and infecting them in a controlled way um, with the viruses that we're interested in. They then become very infected, and we can use them to harvest huge amounts of viruses that can be used for these infection experiments. And we do this traditionally by opening the caps with a pair of forceps, carefully removing a pupa. As you might expect, this is really, really difficult and takes a really long time to get very many. Um, but we figured out a way in collaboration with a few other people to do this in a much faster and easier way. And to do this, we go out into the field and we collect a frame where there's lots of bee larvae that are not quite capped, but will be capped within about 24 hours. We then brush up all the bees and bring them into the lab. Now, these bees, again, they're not capped. They still were going to get fed probably one more time before they were capped. And so instead of putting them in with bees, we just place them into a tub face down, the frame on its face, and we put them in an incubator with the right temperature and humidity. Now, the larvae, once they start to get hungry and no worker um, comes to feed them, they appear to still have this instinct to search for food, and they'll crawl out of the cells and they'll fall out. Again, in nature, this would not happen unless there were no bee larvae or bee adults to take care of the larvae. But it's still an instinct that they have. And so we can use this to get the bees to all crawl out on their own. And we can get a huge number of bee larvae this way. And so you can see these bee larvae are all totally fine. They're alive. They've fallen out onto these special paper towels that we use. And so we can use this as our starting material and take all these larvae without having to do very much work. After they all crawl out, we simply just sort them out onto a tray like this so that they're not touching each other. So it's easier for us to count them and clean them. We do remove any that are really sick looking. We then put them back in an incubator where they can then mature. One thing that you'll see on this, so this is a tray that has had those larvae start to pupate. One of the things that you can see here is lots of little dots of this meconium, which is the last defecation they do right before going into a pupal stage. Many of you might know meconium if you've ever had a baby. It's that first feces, that first defecation that babies do. It's really dark, like black in color, and it's different from their future defecations. Honeybees do something pretty similar. The larvae don't normally defecate; they just eat and eat and eat and eat, and then only right before they pupate they release this meconium, this defecation. And then go through pupation. Normally, this is just at the bottom of the cell. The worker bees clean it out. Of course, there's no worker bees, there's no cell, and so we can see it here. There's a liquid component to it that just absorbs into the paper, and so we can see that waste here. It just absorbs in the paper and doesn't hurt them at all. And so these bees, we let mature until they get to the right stage. They will mature all the way to adulthood. Um, but we can also use them to inject with viruses and then harvest these virus particles for the experiments that we do in the lab. This advance to kind of use the bees' natural instincts to be able to remove large numbers of larvae has increased the speed at which we can do this by about tenfold. It's much easier, it's much more repeatable. It doesn't require as many specialized skills to pull them out. And so it's been a big help to us in a lot of our experiments that require us to use viruses. So that concludes our tour of the bee facility and the lab space that we use to do the research in my lab. I hope everybody found it interesting and informative. As I said, instead of focusing on the results of what we're doing, I thought this would be a fun opportunity to show how we set up these experiments. When you see news headlines or you read summaries in magazines like American Bee Journal and Bee Culture, you often see descriptions of scientific experiments that are done, but it's not always clear how those were set up and how they're performed. And so I thought this would be a nice chance to show off some of those things that are really hard to show off in a real physical tour because we can't let a lot of people into those incubator rooms or into some of our lab spaces. And so this video lets us show those things and kind of make the most of some of the challenges that we're facing in having these types of presentations right now. And so I hope that everyone has a productive beekeeping season and a nice summer overall. We're going to do our best to try to see how much work we can really get through. Luckily, the bees don't seem to mind. And so we still have a lot of good research that we can do this summer, even given the measures that we have to take to make sure that the staff and students here at the University of Illinois remain safe. So thanks again for your attention, and I hope to have a chance to present to ISBA again physically in the near future.